It really amazed me how these two green budgies could end up with this color diversity in their offspring. Look at that. This one is green, as expected. This one is yellow with some green areas. Well, I can understand that. But what about this one? It doesn't have a single green spot in its whole body. What? But wait, there's more. Are you ready to get shocked? This one is blue. Holy mother of Jesus, how? I was baffled until I discovered the basic foundations of Budgies genetics. It all made sense. It's obvious they would turn out to be colored in this pattern if I just followed the basic foundations of genetics. Did you know Budgies don't have the green color gene? Oh, you didn't? So come and join me in my detective journey, where everything is trackable, nothing gets erased, and all is being discovered by the power of genes. The blueprint of our every single trait and feature, where you know more about the parents from their children and about the children from their parents, where nothing can be hidden. In the wild, Budgie parakeets are all green with yellow, with black stripes and markings, and dark blue-green-black flight and tail feathers. We call it the wild type. In the wild, there are no other varieties or color deviations, because any mutation in colors will cost wild budgies their lives. However, pet budgies have a totally different story. They don't need any camouflage to survive. And any mutations in colors for pet budgies does not pose any threat on their lives whatsoever. That's why nowadays we can see a lot of different colors and varieties in budgies that it takes us so much time to decide which budgie we want to take home with us. A budgie has 2 copies of each gene, one from each parent. The phenotype is the physical expression we can see, and it can hide a lot of traits a budgie may have. We can see 2 green budgies having a blue chick. Isn't it amazing? How is it possible? Well, in order to understand that, we need to look at the genotype, which is the set of genes in the DNA that are responsible for a particular trait. Budgerigars are classified according to color and variety. The color describes the general plumage of the budgie, and the variety refers to the pattern and or color of its marking. All colors you see in budgies are caused by 2 genes, melanin and psittacine. Melanin is responsible for all the dark colors you see: brown, gray, black, and even blue, whilst psittacine is responsible for the yellow pigmentation. All budgies are grouped into 2 base color groups: the yellow-based color and the white-based color, which happens when the psittacine gene doesn't work. When you add the color blue to the white-based budgies, You can see all the different varieties in Budgies, but with white and blue. When you add the blue color to the yellow-based Budgies, you can see how mixing blue with yellow gives us the green color. When the Psittacine works, all the varieties you could see in the white-based color Budgies will turn into yellow and green instead of white and blue. Exactly did my 2 green Budgies give rise to 3 yellow-based Budgies and 1 white-based Budgie? Who or what is responsible for the Psittacine to be on or off? Let's refer to the gene that is responsible for the green color as BL with capital B, that signals to the Psittacine to work, and the gene for the blue color as BL with small b, and it shuts down the Psittacine. BL as an abbreviation for blue. The reason we refer to both of them with the gene BL or blue, because it all depends on the Psittacine if the bird is green or blue. As we already know, but the melanin gene itself always produces blue color. The green gene for both parents is BL, which means the Psittacine for both parents is working. But wait! When we give 2 BL genes, we end up with green Budgies. How did we end up with a blue one? Don't forget, each parent has 2 parents as well that got his or her genes from. So both of the parents have BL genes that they got from their parents. These 2 genes together decide if the Psittacine works or not. Now we understand that the 4th child has his Psittacine gene turned off. But if both parents have BL genes, where did their son get the BL genes that turned off his Psittacine? Here's the thing, his parents could have BL with BL genes and still make their Psittacine work. We say the green gene is dominant over the blue gene, or the blue gene is recessive to the green gene, and that's why they both look green. Because the green gene is stronger than the blue gene and didn't let the blue gene to show off. In order for the Psittacine to be turned off, we need 2 BL genes. Otherwise, the BL with the capital B will show off as the phenotype. If a Budgie has 2 different forms of a gene, it is called heterozygous for that gene. As in this case, both parents are heterozygous for the green gene, and the dominant gene takes over the recessive one. So the blue gene is kept hidden from us until finding the opportunity to rise again. As in the 4th child, which has 2 of the same gene, and it is called homozygous. because both of his genes are BL with a small b. In order to understand how two green birds gave rise to a blue one, we need what's called the Punnett Square. It helps us calculate the probabilities for each combination of the parents' genes. When two budgies breed, each parent gives one of its genes, so the children would get two genes in total. We split the father's genes on one side of the Punnett Square and the mother's on the top side of the Punnett Square. And now we join each individual gene with one of the other parent's genes and see all the probabilities. Since we know the BL with capital B gene is dominant over the BL with small b gene, we can calculate what percentage of the offspring will have his psittacine turned off. And it is only when the two small BL genes combine. And we know when the psittacine is turned off, it gives rise to the white series budgies, or as we see them, blue budgies. Looking at the Punnett Square, we can see 75% of the offspring will be yellow series budgies, and 25% of them will be white series budgies. Each gene can have the original form, the wild type, and one or more mutated forms. In our case, the BL, which is the green, is the wild form, and the BL, which gives us blue, is the mutated form. If a budgie is heterozygous for a particular gene, the one that the budgie looks like is said to be dominant, whilst the other, which you cannot see, is recessive. A heterozygous bird is usually referred to as dominant gene split recessive gene. For example, green split blue, which written as green/blue. Each gene is named after the effect of the mutated gene. Each gene is represented by a symbol, usually the first letters of the gene's name. For example, BL, which stands for blue. The dominant form of the gene is written in uppercase. The recessive one is written in lowercase. Now we can have a better look at the Punnett Square, and we can see that 25% of their offspring will be homozygous green budgies, 25% homozygous blue budgies, and 50% are heterozygous dominant green split recessive blue. If one of the parents has a homozygous blue gene with capital B, or let's call it green gene, and one has homozygous blue gene with small b, or as we call it blue gene, let's see what happens to the offspring according to the Punnett Square. We can see all their offspring will be green budgies, and all of their offspring will be heterozygous dominant green split recessive blue. In simple terms, all their children will be green budgies, but all of them carrying the blue gene and could have blue budgies in their offspring. Of course, depending on the other parent. Now we know BL gene is the one telling us if the budgie is white or yellow based. But it is still a long journey from the moment the BL gene gets secreted from the melanin until it reaches the feathers of budgies. You can imagine a tunnel filled with genes that all are awaiting their turn to put their signature on the color we see. We have the gray gene, which is dominant to the wild type, and if present, it dyes the color with gray. And since it is dominant, that means one capital G is enough to leave its signature on the color. Then comes the dark gene, and this gene is greedy. The more of this gene is present in the tunnel, the darker the budgie will be. That's why we have 3 color varieties of each base. In the yellow base, it gives us light green, dark green, and olive. This is how it looks in the Punnett Square if we bring 2 dark green parents. As for the white base, it gives us sky blue, cobalt, and mauve. This is how it looks in the Punnett Square if we bring 2 cobalt parents. Then comes the violet gene, and it acts like the dark gene, greedy. The more the violet gene present in the tunnel, the more violet the budgie will appear. Since we know budgies can have a maximum of 2 from each gene, and this gene is dominant over the wild type, then if one of this gene is present, we call it single violet factor. And if 2 are present, double violet factor. And if none is present, then we call it normal, as it is how budgies are in the wild. This is how it looks in the Punnett Square for the violet gene. And it is very similar to the Punnett Square for the dark gene. And finally, at the end of the tunnel, there comes the Dilution Gene. It is recessive to the wild type. You can think of it as a layer protecting the tunnel, and the more of this gene is present, the more protected the tunnel is. Protected from getting diluted with the base color of the budgies, which opens for us a lot of color deviations that we have to classify these budgies with different varieties. And varieties are a whole new chapter that gives rise to what we see in today's colorful pet budgies. Take a look at the Eno gene, for example. This gene prevents the work of melanin, and we end up with all the genes present in our budgies, but they cannot express themselves, because melanin doesn't secrete anything to the tunnel. And only the psittacine could work, which gives us a Latino, a totally yellow budgie, or if the psittacine doesn't work, gives us the albino, totally white budgie. These budgies have a lot of hidden genetical information in them, but because the melanin doesn't work, it doesn't secrete anything to the tunnel, which makes us wonder what genes are hidden in their tunnels. They could have a very beautiful children with a lot of different colors, but all are hidden inside. There are a lot of amazing varieties for each group, the yellow-based and the white-based groups. But this is a story for another time. As for now, Chapter 1 is completed. But it has opened a new sea of genetical information to discover about our beloved Budgies, with a lot more colors and varieties to come. BudgieWorldwide.com